The first one that we can think about is we're trying to capture some form of motion relative to a stationary camera. And the key thing with this is that the motion might not be predictable. It might be, in various directions with various velocities. We might be trying to capture that motion and characterize that motion, but in general, we can't really predict what's happening with the motion, and we're just trying to capture it as it occurs. The next three jobs are somewhat different. We have, on the one hand, ongoing live analysis, where we might have a flow cytometer again, where we've got cells flowing past a stationary camera, and we're maybe trying to extract some data about those cells, and the image of the cell is not necessarily the primary output. It's something that has come out the other end of analysis pipeline that's our primary output. But here, usually, the motion is somewhat inherent to the technique of imaging that we're using. Similarly, if we're trying to do tile and stitch, again here, motion is inherent to the technique. So in this case, of course, the yellow box shows the field of view of a single camera frame, and we're moving our stage or a gantry if it's larger scale objects that we're trying to image. We're moving our stage from place to place and stitching together a larger scale image. And finally, we might have some uniform motion, again, such as a microfluidic cell or flow web inspection or whatever it might be. And we're trying to capture a movie or capture single frames or whatever it might be of something moving with relatively constant, relatively uniform motion past a stationary camera. And this has to be treated slightly differently from this first job, and there's some other interesting opportunities with this type of job, which we'll come to later.